Salam Sejahtera. Today I would like to present my final year project 1 entitled Growth Efficiency of Endophyte Marasmius Cladophyllus UMAS MS8 on different leaf litter. These are the components of my presentation slide. Start with the introduction. Fungi is a plant litter decomposer that able to degrade lignin and also other components in the plant litter. The higher decomposition rate will cause the decreasing in the litter mass. So here is a various type of media that can be used for fungal growth. Selecting significant media for the fungal growth is very important as it will influence the ways of evaluating fungal growth. Marasmius species, including Marasmius Cladophyllus, are widely found on lignocellulose containing dead plant such as leaf litter, wood, and trees. The fungal growth efficiency is associated with the growth rate of fungus. This means high growth rate of fungus shows that efficient growth and development. The problem statement of this research is, aside from previous study on Endophyte Marasmius Cladophyllus UMAS and MS8 that done by Niang et al. 2017, Fatin et al. 2016, Nur Adila et al. 2017, and Niang et al. 2017, the study of this Marasmius Cladophyllus growth is still lacking, since these studies are mostly focusing on the dye discoloration and lacase production. Then, the hypothesis of this study is fungal growth efficiency is influenced by the capability of fungus to adapt with the provided substrate. So, the objective of this study is to obtain fungal biomass and radial growth of Marasmius Cladophyllus grown on both solid media and liquid media supplemented with leaf litter. Second objective is to identify the suitability of selected leaf litter as substrate of Marasmius Cladophyllus growth. The first literature review is agricultural waste as growth substrate. The growth substrate with different property can influence the moisture accumulation that promote mold growth. Fungi can be used to convert and recycle the agricultural waste such as wood pellet, wood chip, and rice straw into a growth media according to the suitability of the fungus itself. The study by Ristiato and Segesti on 2015 found that Marasmius species cultivated well in oil palm empty fruit bunches and able to reduce lignin by 35.94% in the time period of 30 days. Besides, the study by Gusmitiati et al. On 2020 found that Fusarium species has the highest percentage of mycelium growth and also have the highest average of the decomposition rate with the treatment of mahogany leaf litter. Next is white rot fungi. White rot fungi is a basidiomycete and play roles as the decomposer of that plant. White rot fungi have been the only fungi that able to breaking down all polymers that discover in lignocellulose. After the degradation process, wood residues have different texture and color based on the species of decay. White rot fungi are divided into two subtypes, which are subtype 1 and subtype 2. Subtype 1 is a class of a corrosive white rot fungi. Meanwhile, subtype 2 is a class of successive or selective white rot fungi. White rot fungi have unique characteristics which are extracellular enzyme system and production of hydrogen peroxide system. This unique characteristic made that white rot fungi able to degrade lignin. The third literature review is about Marasmius Cladophyllus UMAS MS8. Niang and colleagues successfully isolate this endophytic fungus from a medicinal plant known as sendudok plant. Through the research, Marasmius Cladophyllus was able to decolorize several types of synthetic dyes, including Remazole Brilliant Blue R dye. This ability showing the capability of Marasmius Cladophyllus to produce a degradative lactase enzyme. Nur Adila and colleagues stated that this endophytic fungus possess potential to be used in the enzymatic biodegradation or microremediation textile effluent. Next, solid-state fermentation. Solid-state fermentation is a fermentation method that occurs in the minimal amount of free water on a solid matrix. This method using solid organic material as a substrate without any pretreatment and creating a natural environment for microbes to grow. Normally, solid-state fermentation used to produce metabolites including enzyme, organic acid, biosurfactant, and more. The productivity of solid-state fermentation is depending on the microbe, solid substrate, bioreactor, and other parameters. Therefore, it is necessary to build reactors that regulate significant aspects of the process. The fifth literature review is related to liquid or submerged fermentation. This fermentation method requires a large amount of water, which is totally opposite to the solid-state fermentation. Submerged fermentation given a high rate of substrate consumption that needed a constant supply of substrate. Most massive enzyme production use this method due to the superior handling and monitoring. Therefore, bioreactor for submerged fermentation has specific features that monitoring and control of parameters. There are four types of fermentation technique of submerged fermentation, which are batch culture, continuous culture, fed bed culture, and perfusion-based culture. The application of those techniques is determined by the selected microbe and final product. The material used in this research is MS8 agar, Marasmius Cladophyllus UMAS MS8, leaf litter of Mimosop elengi, Cinnamomum iners, and Felicium decipiens. The first method step is leaf litter collection and processing, where the sample of leaf litter will be collected from area around UMAS. Then the leaf litter will be processed into a powdery form. Next is fungal growth testing on solid and liquid media. The culture media will be prepared with addition of leaf litter and also minimal growth support nutrient. The only difference between solid media and liquid media is solid media is prepared with agar-agar powder while liquid media is not. For fungal inoculant preparation, stock fungus will be prepared on a mild extract agar. Then the fourth step is culturing test fungal by cultivate it on a solid media and liquid media containing leaf litter. When cultivating the fungal on solid media, two straight lines should be drawn perpendicularly in the midpoint of the agar plug as shown in the picture. Lastly, the analysis of fungal growth. This step will compose of biomass production measurement and also observation of weekly radial growth of fungus on solid media. The picture shown that the biomass production that have been dried on a filter paper. The expected outcome of this study is the adaptation of Endophyte Marasmius Cladophyllus UMAS MS8 that culture on media containing selected leaf litter able to be determined. Then the best leaf litter for the growth of Marasmius Cladophyllus will also be determined. The most important point is this study may provide accurate and up-to-date data on Endophyte Marasmius Cladophyllus UMAS MS8 either for educational purposes or industrial application. These are the work schedule for this study, where the proposal writing has been done on October, November, and December of 2021, while the proposal submission will be submitted on January. Start from February until June of 2022 is used to completing the FYP2. That's all from me. Thank you.